Alright guys, so keep in mind that you should be working on page 102 right now. And I'm going to go ahead and get suited up.
Suited. Transform. No.
So that we can do what we need to do. Yesterday we did have the online Teams tutoring. I think 4 people showed up for that. So keep that in mind, because as we near the end of the 9 weeks, which is next week, and some of you start actually paying close attention to what your current grade is in the class is, you're going to start sending me those emails that say, what can I do to bring up my grade? To which my response will be, try harder next 9 weeks, because built into this 9 weeks, you have had extra credit opportunities very few have taken advantage of. You have also been afforded the opportunity to do online tutoring on Tuesdays and Thursdays from 4:30 to 5. And very few have taken advantage of that. There are, of the 4 people that show up, I think 2 come from this class, if I'm not mistaken. I know Araya's always there. Huh? You were supposed to, yeah, Caleb told me, but you didn't log in. What happened?
Miss Young, you make it seem like I come every time.
I haven't had that many.
No, I don't. I ain't booked Thursday. Miss Young, I can't come on Thursday.
That's fine. That's fine. I mean, you're not required to come at any time, guys. It's just something that is available to you to help you out, since we don't have math break and before or after school tutoring. You can do it from your phone or your computer, wherever you are. You can just sign in to Teams and join in for that if you have a question or if you just want to hear what I'm going over. Again, it is not required, but it has been made available. So before you send that email requesting what can I do to bring up my grade, I just want you to be aware of how that response is going to go if you are someone who has not attended any tutoring nor attempted to do any of the extra credit that was afforded to you via Khan Academy. Yes, Miss Shania, you have a question? There is one this upcoming Saturday, because I realized that the A-Days test is on Tuesday, so it would not benefit them at all to do a tutoring on a Tuesday after they've taken the test. So I will on from 2 to 2:30, I think it is, or is it 2:30 to 3? I have to look. It's 2:30 to 3 on Halloween. I will do a session then. But typically I'm not doing them on Saturdays, because the idea of having to get up and look presentable enough to be on a camera for students on a Saturday is stressful. All right.
So what happened on Wednesday?
Nope. Nope. Your test is next Thursday.
Next?
1 week from today. All right. So, yes, this test is the last major grade for this 9 weeks. And then I think it's the week after. I have to double check the dates. You'll have your Case 21 test, which is the district test. And that'll be the first test grade for the second 9 weeks. I'll have to double check the dates on that and get back to you. But the district test, all biology students with the same test. Any student that is enrolled in Biology 1 in Madison County School District will take a Case 21 test. And you'll do that each 9 weeks. It'll actually be your semester exam for the semester, and you'll do 1/3 9 weeks. Fourth 9 weeks, you don't have a district test because you have the state test at the end of fourth 9 weeks. All right. So anyway, hold on. Is this about this? What's up? You sure? All right. So, what we're going to do, did we start on page 102 last class?
No.
No, we did not. Did I assign you your organelle, though?
No.
No? Okay, cool beans. All right, so this is the way we're going to do this. We are going to go straight down the column. And so I'll do cell wall, so you'll have an example of how we'll do it. And I'm about to call your name and assign an organelle to you so that when I say the organelle, you will be the one that will respond for that organelle. No one else should be responding for that organelle. So I'm doing cell wall. So that means cytoskeleton goes to JC. No. Going in order. Cytoskeleton goes to JC. Parker, you have cell membrane. Caleb, you have cytoplasm. Sean, you're doing ribosomes. Elijah's testing. So Cam, you're doing Golgi apparatus. Muhammad, you have smooth ER. Paige, you have rough ER. Zion, you have the nuclear membrane. Araya, you have the nucleus. Noah, you have the nucleolus. Elisha, you have mitochondria. Eli P, chloroplast. Eli R, lysosomes. Micaiah, centrioles. Christina, centrosomes. Shania, cilia and flagella. Oh man, if I do that, so Bay, you get plasmid. Bo, you gonna have to do cell wall. I'll do the first part of cell wall, to kind of get you guys going. But I forgot that this is my largest class, so it's not enough organelles for everybody to get one if I take one.
I can do the cell wall. He can do mine.
Nope. You're doing smooth ER. He's doing cell wall. All right. So this is how this works. When I say the name of the organelle, we're going straight down the column. The first column, which you're going to tell if this organelle can be found in prokaryotic, eukaryotic, or both types of cells. Okay. So you'll either say pro or prokaryotic, you or eukaryotic, or you'll say both. That'll be your answer. Everyone else should be writing, but after I confirm the answer is correct. So as soon as they say it, don't write it. Wait till you get confirmation from me that lets you know that answer is correct. All right? We're going to go down the column first for prokaryotic and eukaryotic. Then we'll move on to the kingdoms, okay? Now, so we have the type of cells first, and then we have the kingdoms. And I want you to keep this in mind while you're doing the kingdoms. It can only be found in bacteria if it is found in prokaryotic cells. So if you have an organelle that is not found in prokaryotic cells, then it cannot be found in bacteria because bacteria are prokaryotes. Okay? Now the other three kingdoms are eukaryotic kingdoms that we're looking at: fungus, plant, and animal. So we don't really go in detail with fungus, but here's a way that can help you figure it out. If it doesn't say plant only, or if it doesn't say animal only, then you include fungus.
What about this stuff? Protist.
I didn't put protists on there. Yeah, protists is not on there. We're only looking at 4 kingdoms. 4 of those kingdoms we're looking at: bacteria, fungi, plant, and animal. Okay? Those are the 4 we're looking at. All right. So, for instance, if I said cell wall and we're doing prokaryotic and eukaryotic, Bo would say both. And that's correct. So then you would write both in the box under prokaryotic and eukaryotic. Everybody needs to write these down on this page. Close your MacBook, love. We're not using MacBooks today. All right. Are we clear?
Yes, ma'am.
Do we understand?
Yes, ma'am.
All right. So we're getting started. Cytoskeleton. Try again. Try again.
Could I have that?
Eukaryotic. Alright, cell membrane. Everybody has an organelle, Muhammad. Stick to yours. Cell membrane. Both. Cytoplasm. Both. Ribosomes. Try again. Keep in mind you have these, all of these organelles are listed on pages 99 through 101. And so if you look at the name of the organelle, it pretty much kind of helps you figure out where it should be found if you look up the organelle. Both. There you go. Both. Golgi.
Eukaryotic.
Eukaryotic. Smooth ER. That's you, Muhammad.
Oh, both.
Smooth ER.
Oh, once again, it's me. I mean, eukaryotic.
Eukaryotic. Rough ER. Eukaryotic. Nuclear membrane. Try again. Eukaryotic. Remember, nuclear membrane surrounds the nucleus, and prokaryotes don't have a nucleus, so they can't have a nuclear membrane. All right, nucleus. Eukaryotic. Nucleolus. Eukaryotic. Mitochondria. Eukaryotic. Chloroplast. Eukaryotic. Lysosomes.
Both.
Try again. Eukaryotic. Centrioles. Eukaryotic. Centrosomes. Eukaryotic. Cilia and flagella. Both. Plasmid. Prokaryotic. Cool beans. I'm going to go back through that to make sure everybody has that written down. So check yourself. If you miss one, now is your chance to put it back in. If I'm going too fast, just say, Miss Young, más despacio, por favor. Oh, wait, it's not Spanish. Slower, please. All right. So, yeah, I don't speak that language. All right. Cell wall, both. Cytoskeleton, eukaryotic. Cell membrane, both. Cytoplasm, both. Ribosomes, both. Golgi, eukaryotic. Smooth ER, eukaryotic. Rough ER, eukaryotic. Nuclear membrane, eukaryotic. Nucleus, eukaryotic. Nucleolus, eukaryotic. Mitochondria, eukaryotic. Chloroplast, eukaryotic. Lysosomes, eukaryotic. Centrioles, eukaryotic. Centrosomes, eukaryotic. Cilia and flagella, both. Plasmid, prokaryotic. Alright, now we're moving on to the next column. This is where you're going to tell the specific types of cells. So whether it's bacteria cells, fungus cells, plant cells, or animal cells. So you will name them, or if it's all of them, you would say all. Okay? So, for instance, Bo will tell me which types of cells specifically have a cell wall. Not all of them. Because remember, animal cells are in there, and do animal cells have a cell wall? No, they do not. So what are the cells? Bacteria, fungi, and plant, to which you will write BFP. That way you don't have to try to spell out all three of those words. You would write BFP. Remember that cell walls, Muhammad, I need you to cover, love. Remember that cell walls can be found in all types of cells except for animal cells and animal-like protists, okay? But fungus-like and plant-like protists would also have cell walls. So each kingdom, except for the animal kingdom, contains cells that can have a cell wall. All right, moving down. Cytoskeleton. Try again. Now, you said all. Keep in mind, you already said it's not found in prokaryotes, so that automatically knocks out bacteria. Because the only way you can say bacteria is if it's found in prokaryotic cells. Because prokaryotes are bacteria and bacteria are prokaryotes. Okay? So if it is not found in prokaryotes, it cannot be found in bacteria. So that automatically knocks out the B. So what types of cells are they found in? Not plants. Fungi and animal. Fungi and animal. All right, coming down, cell membrane. All of them. Cytoplasm. All. Ribosomes. All. Good job. Golgi.
Planet, fungi,
And
animal.
animal. That's right. Fungi, plant, and animal.
So it's not all.
Not all,
All
because
would
remember
be
it's not
bacteria,
bacteria.
too.
Right. Right. You were right to not say bacteria, but you forgot animal. So it's fungi, plant, and animal. Good job. Smooth ER.
Plant, fungi, animal.
There you go. Fungi, plants, animals. All right. Rough ER. There you go. Nuclear membrane.
Me.
Yep, that's you. Nuclear membrane. Fungi, plant, animal. All right. Nucleus.
I guess mine or something.
I skip you.
I don't know.
This here.
You had Golgi?
No.
I had goats.
Okay. We did smooth and rough, nuclear membrane. Now we you got nucleus. Making me think I'm crazy. All right, nucleus, a little crazy self.
Fungi, plant, and animal.
Fungi, plant, and animal. Nucleolus. Fungi, plant, and animal. Mitochondria. And there you go. Fungi, plant, and animal. Chloroplast. Plant. Lysosomes. Nope. Remember we already said it's only found in eukaryotes. It's not found in prokaryotes, so it can't be found in bacteria. And lysosomes is one of those organelles that's a only situation. So if you look under vesicles, which you can find on page 100, and find lysosomes, it tells you specifically where you can find lysosomes. So where would I find lysosomes?
You would find.
It's not yours. Hush.
I'm being a good
Look turn to page 100. Find vesicles, and under vesicles, you're going to see lysosomes. And tell me what it says under lysosome. The word lysosome, what does it say under it?
Which one?
Where's Leslie?
Lysosome.
Oh, 100.
Hush. Eli, come on.
You got it.
The word lysosome is on page 100. If you turn to page 100, and there was nothing you had to fill in, it was already typed there for you. If you turn to page 100, if you look in that third row where the vesicles are, the last organelle listed in that third row is lysosomes. Animal, right. Lysosomes are animal only. Lysosomes are animal only. Some scientists believe that they may be in plants, but you can't see them. So since we can't see them, we're not going to count that. It's animal only. All right, centrioles. Try again. Animal only. Uh-oh, I didn't mean to cut you off. I thought you were just going to say animal. I'm sorry. Centrioles are animal only. Turn to page 100. And look where it says centrioles are found in eukaryotes. It's in the last column. Centrioles are found in eukaryotes. And what does it say right under that?
Animals.
Animals. All right. Centrosomes. And there you go. Fungi, plants, and animals. Let me explain to you the relationship between centrosomes and centrioles. Everybody look up this way. Give me your attention. All right. They function in the same manner. They're going to aid in cell division. A centrosome is going to function in cell division. It aids in cell division. If it is found in an animal cell, there is something on the inside of the centrosome, and the something on the inside is called a centriole. Okay, so centrioles are located inside the centrosomes of animal cells.
Oh.
Centrosome of plant, nothing on the inside. Centrosome of animal, centriole on the inside. Got it? They both aid in cell division, but animals have the centriole because the centriole creates the band that helps to cleave the cytoplasm during cell division. Plant cells don't cleave their cytoplasm during cell division, so they don't need something to make the thing to cleave the cytoplasm. Are you with me? Centrosomes, both. If it's in an animal cell, there's a centriole on the inside of the centrosome. All right. Get back. Stay where you're supposed to be. Stop moving. Talk to the microphone, not y'all. All right. It might be doing its own thing sometimes. All right. Cilia and flagella. All of them can be found in all of them. Does that mean that every cell that is an animal, plant, or fungus cell has cilia or flagella? No, but you can find some cells in animals that will have cilia and flagella, okay? Plasmid. Bacteria.
Good job.
All right, awesome sauce. Now, I am going to give you 2 minutes. 2 minutes. And it'll start when I say start. You will have 2 minutes to look up the function of your organelle and write it on your line. You can find the functions between pages 99 and 101. So that means put your phones up. You are not Googling. You are reading from your notes. Between pages 99 and 101, and your time starts now.
We're not going over it?
Yes, we are. That's why I want you to find yours so that when I call for it, you'll already have yours so everybody else can get it. Yes, ma'am. Have you already broke down your function? Can you be back within 2 minutes? Go for it. She beat you to it, Bo. Yes, ma'am.
What if it doesn't say
We went over it the other day. So which one do you have?
That's what I did.
It does. It says it at the very top of centrosome. It's the first 2 words. And I just said it out loud a few minutes ago. Centrosomes are on page 101 at the very top, the first column. You missed it?
What about my smooth ER?
You better know what smooth ER do.
I know what it is, but I'm just making
You want to know what page it's on?
Yeah, make it sure.
It's on page 99 at the bottom.
So we still have to fill them in.
We went over these the other day. Yeah, you should have already had it filled in. Why you got your whole mouth and nose uncovered. Do you know the numbers are rising? Rise your mask. All right.
Me no English. Put that mask.
Okay. Well, me speak fight. Put up mask.
Are we gonna go like over if they are in bacterial plants or anything like that?
What?
Are we gonna go over like we did on the?
Yeah. We're gonna go over it.
Look, it's falling down. Say the one step.
Okay, you need to twist the thing around the ear. Like take the thing out, twist it one time, put it on your ear to hold it up. It'll make it tighter.
It hurts my ear.
Oh, it's gonna hurt your feelings if you go look over there one more time with the mask down.
Oh no.
Me and you gonna fight. I'm gonna take one of my, you know I got these rubber bands that if I trusted to hold my iPad, I'm gonna put that rubber band around that mask in your head and trust to hold your mask up on your face. Don't play with me.
Me no English, me no understand.
Okay, but you speak movement, right? You understand if I come over there and do it. Don't play with me. You speak perfect English. Your English better than mine sometimes. It's open. You accurately conjugate your verb stuff. What? Yeah. I was waiting on you. You was late, but I was waiting on you. You weren't waiting on. Alright. Let's get ready to go. So just like we did everything else, I'm gonna call your organelle and you are going to say the function. Now, I may ask you some additional questions. That does not mean that your function is incorrect. I just want to make sure that you understand what you're saying the function is. I don't need you to just memorize some words. I need you to know what that thing means, okay? So, for example, If I said cell wall, Bo would say that would be the cell membrane. Cell wall. What's the function of the cell wall?
No, it's supposed to.
Say that again. Oh, I was waiting on. Is that you, Bo, talking to me? Oh, so you sounded different all of a sudden. So it provides structure and support for the cell. Structure and support.
One second.
Structure, support, protection would be words that you would want to put there for the function of the cell wall, right? Structure, support, protection. All right, so cytoskeleton.
Support and function.
You are smooth ER. That's all. Smooth ER.
Okay.
Smooth ER.
Okay, for the cell wall.
Not rough, but smooth. ER. That's you. All right, cytoskeleton. Let's go. Structure and support. For what kind of cells? Not all of them. Remember we just said what kind of cells is found in. Right, there you go. So this, guys, y'all got to put this information together, okay? So all of this goes together. All right, so structure support, and it can also help with movement. It can also aid with movement. All right. Cell membrane.
Like filters out the stuff and like protection and structure.
Not protection. Protection is not the cell membrane's job. So what does the cell membrane do? Come on, talk to me. I can't hear you.
Okay. It like when the cells are coming in.
You know it's on page 99 at the top. It's the first one.
Give them a better.
Hush, woman. I mean, man. I'm sorry. I'm so used to talking to females. I'm sorry.
It regulates the cells.
Go to page 99. Look at the last box and tell me what that says. The last box in the first row, I should mean. Because those are the functions.
Cell division.
Okay. Find cell membrane. Do you see it on page 99 at the top? Do you see cell membrane? It's the first organelle.
It's on the one before the last box.
Now, the functions are in the last column in that row. So tell me what's the function of the cell membrane.
Regulating cell transport.
It regulates cell transport. So it maintains homeostasis. Maintains homeostasis by regulating cell transport. And what that means, guys, because a lot of times students will say it maintains homeostasis but not know what that means. Homeostasis is having a stable internal environment. So the cell membrane makes sure that the internal environment of the cell stays stable, and it does that by determining what can enter and exit the cell. That's what it means by regulating cell transport. So when you see maintain homeostasis by regulating cell transport, what that really means is it's keeping the inside happy by letting what needs to go out, and letting what needs to come in. It controls the things that enter and exit the cell. Are you with me? All right, let's move on to cytoplasm. That's where it is, and that should actually edit that. It's a typo there. It should say fills the space between the nucleus and the cell membrane. So all the space between the nucleus and the cell membrane is where you find the cytoplasm. That's where you find it. But the question is, what does it do? Function is what does it do? So it's located between the nucleus and the plasma membrane. Hold on a second. But what does it do?
It helps.
Come on, cytoplasm. That will be right below. It tells you where it is first, and then right below that it tells you what it does. What we are seeing is the result of not filling in the blanks on our notes. And since we didn't fill in the blanks on our notes, we don't know what to say after sight of most, because that is an important word right there. What's that word? Metabolic. What's your question back there? I can't hear you. But that's not the main function. Oh, yeah, it helps with holding the organelles in place. But what we want to know about the cytoplasm is that it is the site of most metabolic reactions, most metabolic activities. So what that means is metabolism is when things are being broken down. So a bigger a bigger molecule is being broken down to its components, or things are being built up. So the components are being put together to make a bigger molecule, okay? So that's what metabolism is. Most of the metabolism, so most of the breaking down and building up that's happening inside of a cell is actually going to happen in the cytoplasm. So yes, it fills the space between the nucleus and the plasma membrane, and it's a jelly-like fluid, so the organelles are sitting in that fluid, so it's kind of like having fruit in jello. It kind of helps to hold them in place, but the main function of the cytoplasm is to be the site of most metabolic activities. That's where the most of your breaking down, please don't write on my table, most of the breaking down and building up is going to happen, okay? I never thought I would have to tell students so much not to write on tables and color my numbers because we were in high school. I thought that left elementary. I thought, that, but it just, it baffles me.
Their brain is
All right.
not processing.
You said you left yours in the hallway, so hush. All right, so anyway,
I thought you didn't find it, did you?
moving on. I didn't. That's why. I said that's what you said you left
Or
it.
somebody stole it.
Hush, young man, till we get to the smooth ER. You better have your smooth ER right while you doing all this talking. All right, next we have ribosomes. Ribosomes. What do that ain't yours? What do ribosomes do, Sean? What do ribosomes do? Ribosomes are found at the top of page 100. To give you a clue, guys, if it says that it's make, if you see a word like make, that's telling you its function. It's making something, okay? Now, you also need to write down, just so you'll know, synthesis means make. Most often on test questions, you won't see the word make. You'll see the word synthesis or synthesize. And so when you see that word, you need to think, oh, it means to make, okay?
Oh.
So when you see synthesis or synthesize, that means to make. So ribosomes, what do they do? What do they make? Not you. What do ribosomes make?
Somebody said
He said proteins?
yeah.
I didn't hear him. I'm sorry. Thank you, sweetheart. That is correct. Proteins.
You are.
Speak up a little bit for me. I'm so used to Muhammad being loud that when y'all speak low, I can't hear you. Gotta put my ears down. I can't turn them down. I'm turn them up because Muhammad be done blew my ear socket out. Well, not an ear socket. That's not an ear socket. I mean eardrum. See that? You make me forget the parts that's in my ear. Whew. All right. Muhammad, you keep me going.
Alright, you tell Muhammad.
Golgi. Golgi apparatus. Golgi. What's the function of the Golgi?
Oh, I said sort proteins and leaves outside the cell.
It sorts. That's definitely it. Sorts, but also make sure you want to write down modify, package, and ship, okay? It definitely sorts. Good job. Sorts, modifies, packages, and ships proteins and lipids that are going to leave the cell. Good job. That was a great job.
Package is what? Shit.
Sort, modify, package, ship.
Package shit.
So it's kind of like, think of it this way. A bunch of stuff comes into the Golgi. It first has to sort it out, right?
Oh.
And then it fixes it or modifies it. It may have to fold it more. And after it modifies it, it puts it in its vesicle so that it can actually ship it out. So it's kind of like an assembly line. You come in, you sort it out, you finish folding it, so you modify it, you package it inside of a vesicle, a transport vesicle, and then you ship the transport vesicle out. So sort, modify, package, ship. That's what the Goji does.
Okay.
All right? And thank you for including that it's going to be for things that's going outside of the cell. Awesome job on that. All right, next we have the smooth ER.
The smooth ER contains enzymes that aid in cell function.
What cell function?
Let me see.
Mm-hmm.
Let's see. The fungi and the plants and the...
Yeah, I ain't said what cell. I said what's the function.
The ribosome and they produce proteins.
No. Ribosomes make proteins. All right. Smooth ER, guys, it does contain enzymes that aid in cell function, but the specific function, this is what I want you guys to write down, okay? It synthesizes lipids. And what does synthesize mean? Make. So it makes lipids and it detoxifies poisons.
Oh.
Those are the cell processes that it does. It has enzymes that help it to make lipids and detoxify poisons. Okay?
So
right.
if you have so much of them, you don't have to worry about poison.
No, you still got to worry. Yeah. All right. Rough ER.
I said it makes proteins.
Does it make them?
No.
Or does it
Help.
transport them? Remember, the ER is a membrane that's used for transport. The reason it's called rough ER because what's sitting on it? Ribosomes. And so what's actually making the protein? The ribosome is making the protein, but it's making the protein so that the protein goes into the ER and then the rough ER transports it. It's called rough ER because the ribosomes are like bumps on that membrane, okay? But the ribosome is actually making the protein. What the ER is doing is actually transporting that protein. I think there's a typo on here, which is probably where you got that from. And I'm making stuff on corrections I need to make on my thing. There's a typo on there. Don't play with me. All right. Next one, we have nuclear membrane. It controls what enters and exits the nucleus. Perfect. All right. Think of it this way. If the cell membrane controls what enters and exits the cell, then the nuclear membrane is controlling what enters and exits the nucleus, right? That's why it's called a nuclear membrane.
What
Cool.
enters the control? What? Mahan, be quiet.
It controls what enters and exits the nucleus.
Oh.
What enters and exits the nucleus.
What enters?
All right, moving on to the next one, we have the nucleus.
The nucleus.
Nucleus, go ahead, Araya.
Control everything in the cell.
So it controls most of the cell's activity. Not all, it controls most of the cell activity. The reason we can't say all is because mitochondria and chloroplasts have their own DNA. So they control their own activities. But the nucleus having most of the cell's DNA will control most of the cell's activities.
Oh.
All right, hush, Muhammad. The nucleolus that synthesizes ribosomes is where the ribosomes are made. It makes the ribosome subunits. Awesome sauce. Mitochondria. It makes ATP. Thank you for going straight there. Love it. Makes ATP. Now, let's put a pin right here and let me explain something to you. The mitochondria, and wait, after we do chloroplast. We'll do a pin after chloroplast. Chloroplast.
Absorb light and make energy.
It doesn't make energy. This is where I want to put the pin. But I see where you're going with. I want to make sure we're clear. We don't want any misconceptions. Energy cannot be created nor destroyed. You cannot make energy. You cannot destroy energy. All energy on this earth comes from the sun in the form of light energy. The mitochondria has the ability to transform light energy into chemical energy. It is an energy transformer. The mitochondria and the chloroplast. Did I say mitochondria or did I say chloroplast? I meant chloroplast. I'm sorry. The chloroplast has the ability to transform light energy into chemical energy. Chloroplasts and mitochondria are the organelles that are energy transformers. That's their job. Energy can be transformed and transferred. It cannot be created nor destroyed. So we can never make energy. Are we clear on that? Wait, now I'm not finished explaining. Hold on. So the chloroplast captures light energy, transforms it into chemical energy where it is stored in the bonds of carbohydrates. Okay? We cannot use the chemical energy when it is in the bonds of carbohydrates. So even though it's chemical energy, it is not usable by the cell. The mitochondria is able to transfer the energy from the carbs to a molecule called ATP. The cell can use ATP. Write this down. ATP is the cell's energy molecule. ATP is the cell's energy molecule. Are y'all with me? Where does all energy on this earth come from? The sun. Which organelle can capture that and transform it into chemical energy? Chloroplast. Chloroplast. All right? That's called photosynthesis. Which organelle can move that energy from the chemical energy in food to the chemical energy in ATP? That's the mitochondria. That's called cell respiration.
The energy in food to...
Okay? So, from sun to food, chloroplast, photosynthesis. From food to ATP, mitochondria, cellular respiration. Y'all with me? All right. Next one.
Doesn't that mean the sun makes the energy?
All energy on earth comes from the sun.
So,
all energy on earth comes from the sun, and we're going to leave it there. All right. Lysosomes.
Disposing of waste.
Disposing of waste using what? How does it do it? You're right. Don't think you're right. So, but we're going a little bit further. How does it dispose of the waste? What does it do? How does it help to clean the cell? That's what it does. It cleans the cell, but how? Using digestive enzymes. Awesome sauce. All right. So we're going to stick a pen right here. I want you to use your imagination with me. And I want you to pretend like I came in here with 3 empty jugs. If there are empty jugs, I can put stuff on the inside of them, right?
Yeah.
So as an empty jug, it's just a vesicle. That's all it is. It's a vesicle that I can put something in. So the word vesicle is a generic term for a small membrane sac that can hold stuff on the inside of it. That's a generic term. Now, I can get specific and give it a specific name based on what I put on the inside. If I put milk into my first jug, then you're going to call it a what?
Milk jug.
A milk jug. If I put water into my second jug, you're going to call it a what? A water jug. And if I put something else in the third jug and whatever, you're going to call it that type of jug. It's still a jug, right? So a vesicle is still a vesicle, but what's on the inside determines the specific type of vesicle. So if I put digestive enzymes on the inside of a vesicle, I call it a lysosome. It's a vesicle. It's a membrane sac. But on the inside, it has digestive enzymes. So because the inside of the vesicle is holding digestive enzymes, I call it a lysosome. If I put water or waste products or salts on the inside, I call it a vacuole. It's a vesicle. It's holding stuff, but it's holding water, waste, or salts, stuff like that. So I call it a vacuole. If I use the vesicle to take something from point A to point B so it doesn't stay in the vesicle, I put it in there, take it to the next point, drop it off. That's called a transport vesicle. Do y'all understand what the term vesicle means? It's a generic term for a membrane sac that is going to hold something on the inside. If I put digestive enzymes on the inside, what do I call that vesicle?
Lysosome.
Lysosome. If I'm using it to hold water or salts or waste products, what do I call that vesicle? Vacuole. If I'm using it to move stuff around inside the cell, what do I call that? Transport vesicle. Do we understand that?
Yes.
They're all vesicles. Yes, sir.
Okay, so why do you call it vacuole and that thing? Like, what's the difference? Like,
Why do I call it what?
Why do you call like a? Isn't like when you said oh, I get it. So yeah, digestion enzyme is another name for lysosomes.
If I put digestive enzymes in the vacuole, I call that vacuole a lysosome. Just like when I put milk in the jug, I call it a milk jug. Okay? So if I put digestive enzymes in a vacuole, I call it a lysosome. I mean, in a vesicle, I call it a lysosome. Okay?
So the same thing as when you put water or...
When you put water, you call it a vacuole. It's a specific name to let you know what's on the inside of that vesicle. So if you hear lysosome, you think, oh, it's digestive enzymes in that. When you hear vacuole, you're going to think, oh, it's water or food or waste in that. Okay? That's all that's for.
Oh.
All right. Centrioles.
Correct the bond that keeps
Okay, so in layman's terms, cell division. Okay, you're right. You're exactly right. No, we just say cell division, but I mean, keep what you got because that's perfect. You went into details. I only have to ask you what they mean. Cell division. That was centrioles. Cell division. What about centrosomes? Cell division. There you go. They aid in cell. That's their job. They help in cell division. Remember, a centrosome in animal cells has centrioles on the inside. So if a centrosome is helping in cell division, then the centriole must be doing it too because it's on the inside. But it's only found in the inside of animal cells. All right, cilia and flagella. Movement, or what we like to call locomotion. Them crazy movements. Cilia got you going like this. Flagella got you going like this. Hey, locomotion. Hey. All right. I know. That's what they keep saying. I don't know why. Ugh, I'm totally normal. All right. Plasmids.
help bacteria survive adverse conditions.
It helps bacteria survive adverse conditions. Right. It helps bacteria survive adverse conditions. And if you don't know how to spell those words, or if that was too fast for you, all you have to do is look on page 101. It is literally the very last thing on that page. Plasmids are the last organelle listed. Now, here's the part where you have to use your brain. Muhammad, it's going to be a problem for you because you say you left yours in the hallway. I don't know what to say. All right, so here you have to think. What is going to be the immediate effect if this organelle is damaged or missing? So you have to look at what it does and then say what will happen if it does not do that thing. For instance, I'm supposed to teach you. If I never come to school, or if I do come to school, I never say a word to you.
Wouldn't have to work.
But will you pass the state test?
Yes.
You think so? With no teaching? Okay. All right then, Superman. Who got the key memorized? What's up? Not yet. When we finish this, you can.
What if you cheat?
Because you are my first person. And you are about to tell us what would happen if the cell wall is missing or damaged.
Oh, like, okay. Would it fall apart? I was thinking it would fall apart, like cells fall apart.
Why would it fall apart? Because what is it missing?
The structure.
Structure and support. Awesome sauce.
So it would fall apart.
I mean, yeah.
But I thought like a waste would come and oh no, right, no, I was thinking for cell membrane.
Uh-huh. Cytoskeleton.
Once again.
What would happen if the cytoskeleton is missing or damaged? It would lose. Okay, so a big picture is the cell would lose its shape and stuff wouldn't be able to move. Okay, the cytoskeleton basically helps. Now it offers support, structure and support, but we're looking more at the structure, the shape of the cell. It's more what cytoskeleton is going to do. So the cell would lose its shape and be unable to move things around. Not the cell wouldn't be able to move. Well, kind of, because it does kind of make. Never mind. All right, so we'll talk about that later. I'm about to get into AP. Nope, not the class. All right, cell membrane.
I'm gonna take a one in.
He said 1 second, 1 minute.
He don't even have cell membrane, so whoever got cell membrane.
I said 1 second.
I said it wouldn't be able to perform homeostasis.
And what does that mean?
They would be unstable.
What does that mean?
He's already said.
Does the game tell you that?
No, it's not a game.
Like they wouldn't be able to survive because because their protection is.
Wait till we get the ER, the smooth kind. Don't make me get rough with you. I can't hear you back there. What is it saying?
It wouldn't be like balanced.
All right, so it would lose its stability. It would lose the internal stability because everything would be able to just go freely in and out how it wants to, right? Or nothing will be able to get in and out. That would be even worse. Let me give you an example that'll help you think about how much worse that would be. What if you couldn't eat or drink anything, so nothing could come in, but you also couldn't poop or pee, so nothing could get out? That's a bad situation, right? All right. Why are you shaking your head, Eli?
I don't know.
All right. That would not be pretty at all. All right. Where were we? Cytoplasm.
There wouldn't be a place for metabolic processes.
All right. There's no place for metabolic processes. So, could stuff that needs to be built up have a place to be built up? No. What about stuff that needs to be broken down? Pretty much no. And also, what about those organelles that's being held in place? They're just going to go everywhere. We're going to have total chaos.
Yeah.
Total chaos.
So
Kind of like y'all when the bell ring and A through K supposed to go first and everybody hop up to leave. Just total chaos. All right. Moving on.
So if one organelle is missing, the whole thing is destroyed.
That's the big picture. It can be,
Okay.
depending on the organelle. Some organelles can be replaced. All right. Where were we? Ribosomes. What would happen if there was no ribosome?
There would be no protein.
That ain't your word. I'm gonna get some tape. Is that legal? All right.
It's illegal, man. It's illegal.
There would be no proteins. There would be no proteins. Why is that a bad thing? Let me explain to you what proteins. Put your hand down. Let me explain to you what proteins do. Proteins function in your immune system, so you would lose your immunity. Proteins function in building up things like your muscles, so you would have muscle loss. Proteins function as enzymes, so there'll be no catalyst to speed up the metabolic reactions. So it would take too much activation energy to get the processes going. Proteins function as a part of the cell membrane to allow some things to get in and out of the cell. So you're going to have reduced homeostasis. Proteins function to allow cells to communicate with each other so that cells know when they should grow, should not grow, when they should do X, Y, and Z. So they could lead to development of things like tumors, which could lead to cancer or otherwise complete and utter chaos. Okay? Proteins, listen to me. The DNA has the instructions for what the cell needs to do. But having the instructions does not mean that you are doing it. For instance, I type instructions for activities on Canvas and in these packets for things for you to do. But when you come in class, you still have not done it. There has to be something to actually do it. So DNA has the instructions, and the protein is the worker that carries out the instructions. DNA only knows what needs to be done for the cell to survive.
Oh.
It sends out a message that says what needs to be done, but it is the job of the protein to do that job. And the ribosome is the thing that is able to make the proteins, okay? All right, moving right along.
So like a coach is telling the boxers to fight and telling him how to fight, and the boxer is knocking that guy out.
Right. The coach ain't in the ring. He said that's actually a good analogy. I know sometimes he be we be in left field and we be in the right field or whatever, or he be outside playing football and we inside playing basketball. I know that, but that was a very good analogy. He said that's like the coach telling a boxer how to fight, but the boxer is the one that actually goes in the ring and does the fighting. That's a great analogy. The coach is like the DNA. It's training, telling the boxer what to do, but the coach not the one getting in the ring. It's the boxer that actually gets in the ring and does what needs to be done. That was a great analogy. Thank you for that one. Alright. I mean, 90% of the time you be on it, but then you get in your rare form and that mouth just starts blah, 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 blah. And I'd be like, look, I'm going to throw something at you if I could. I'm going to hit you with a wet noodle, like for real. Wet noodle, so it just stick to your face and make your face feel dirty all day, but you ain't hurt. Just a wet noodle. Alright. So anyway, moving on. Hush. You had a moment. Let it go. There was ribosomes, right?
Yeah.
Golgi. That ain't you. You can do it, Cam. You told me what the Golgi does, so what happened if it doesn't do that?
Ain't gonna be no energy.
Now, wait a minute, think about Golgi. What does Golgi do? Look at what you wrote down that Golgi do.
Protein and lipids outside the cell.
So if there's no Golgi, then what's gonna happen? So if it sends the proteins and lipids outside of the cell, Golgi's not there. What happens to those proteins and lipids?
They're not gonna come in.
They're not gonna be able to go out the cell. Okay. Now let me explain to you why that's important. Let me give you something that you can visualize. When you start to eat your food, what happens in your mouth?
You eat it, you chew it.
While you're chewing, what is happening? What's the physiological thing that's actually happening? While you're doing the chewing, what is also joining that food? Saliva. So you have salivary glands that are actually secreting enzymes to start the chemical breakdown of that food. That means that the glands in your mouth have a lot of Golgi. Because the Golgi is the thing that is sending those proteins, remember proteins are enzymes, sending them outside of the cells so they can be secreted into the mouth to help break down the food. If there's no Golgi, you don't get those enzymes to come into your mouth to start helping break down that food. Same thing with your oil glands or your sweat glands. Any of your glands are going to be the things that secrete things. The cells that make up those glands are going to have a lot of Golgi bodies in them, because the Golgi is the thing that's going to make sure that the stuff that needs to be secrete means to exit. The stuff that needs to exit the cell is going to get out of the cell. If there's no Golgi, that does not happen. All right, y'all with me? Yes. Imagine trying to eat and you making no saliva. Like, that would be just horrible. Like, ugh. Anyway.
Can we skip the next one?
No. The next one is yours. You've been doing all this talking.
Okay.
All right. Smoothie R.
Okay. Listen careful. You wouldn't be able to have a cell membrane because you wouldn't be able to have lipids.
Okay. You would have lipids. That's a good one. Good job. You did it.
If they poison you, will die.
And look at you.
I'm so smart. I know. Awesome.
You had your moments. All right. Ruffy R.
1 second. Ruff is supposed to wait for this.
Ruffy R.
Proteins get backed up.
They get backed up. Because the ribosome making the proteins, they ain't going nowhere. Just sitting there. Can't even get out the cell. All right. Nuclear membrane. Nucleus does not have any balance. Wouldn't even be a nucleus. DNA just sitting in the cytoplasm like it does in prokaryotes. Wouldn't even be a nucleus. There's no nucleus. DNA is not protected from the cell environment. All right?
There would be no nucleus?
There'll be no nucleus. Nucleus. If there was no nucleus in the cell,
No cell.
it might be a cell. It's going to be a worthless one. Because there's nothing to control the activities. So there's no regulation of cell activities if there's no nucleus, right?
No leader.
Alright, mitochondria. I'm sorry, nucleolus. I skipped one. Nucleolus.
No ribosome.
No ribosomes. That's a real bad problem. Because what do ribosomes make?
Protein.
Proteins. And we already talked about what's going to happen if you don't have protein. So we got Eli. Bruh.
That's kind of sad.
What, I'm listening?
It's okay.
You supposed to be writing though. How you writing?
I got it written down.
Let me see. Bring it here. Ratna. R-A-T-N-A. Bring me that page. Ratna. Come on now. I spoke Ebonics to you. I want to see it. Ratna.
What is phonics?
Ebony phonics.
I still don't know.
Ebony, African American phonics. Alright, what's up?
I got stuff from rough endoplasmic reticulum.
No, you're supposed to be writing down everything as we go through it. That's why we're going through it. Alright, where are we now?
Mitochondria.
I skipped nucleus. No, we didn't.
We said no ribosome.
See how you didn't want to hear me talk.
No, nobody want to hear you talk. What you supposed to
I realize we close to lunch, but we're not at lunch, so let's chill. Alright, nucleus is, there'll be a loss of cell activities. And we did nucleolus, there'll be no ribosomes, so now we at mitochondria, which means there will be no energy that the cell can use. There's no ATP, okay? Because the cell energy molecule is ATP. I shouldn't say no, because you get a small amount of ATP that will be made in the cytoplasm, but the majority of the cell's ATP is made in the mitochondria. So you'll have a great reduction in the amount of ATP. Yes, Mr. Eli.
Whenever you said that energy cannot be destroyed,
Uh-huh.
what about depleting energy? What does that mean?
You have transferred it to somewhere else. Most energy is transferred to heat energy to the universe, but energy is not destroyed. It is either transferred or transformed to a different kind of energy.
So no matter what happened, energy will always stay there.
Right. Energy always exists. It may be like it can leave one thing and be transferred to another thing. Kind of like when a battery is drained, you've moved the energy from the battery to something else. Like that energy has moved, but it's not destroyed. Yes.
What about a flashlight?
What about it?
Like, if it's draining a battery into a flashlight,
And it's transforming it into light energy, and then that light energy is transforming into heat energy. Ever stick your hand around a light, you feel all the heat that's coming off of it? That's the energy being transformed to a different kind. So it's going from stored chemical energy to light energy to heat energy. Most energy ends up as heat. Okay? But it's the energy transfer is one directional, like you can't go back with it. So you have to have a constant input of energy, but you're not destroying it. You're just moving it somewhere else or changing it to a different kind.
that is crazy.
I know, right?
So I see, like me.
You know, my grandma used to always say there's nothing new under the sun, and I guess she was right. It was always here in some type of way, ships.
Maybe
All right, where are we now? Chloroplast?
Yeah.
Chloroplast. No chloroplast.
The plant will not be able to transfer light energy into like usable energy.
Right. So if there's no usable energy, we screwed. Like, we can't go outside and go, photosynthesis, photosynthesis, photosynthesis. And then all of a sudden, like, ooh, I'm so full. Like, it don't work that way. I wish it did.
No.
Okay, that would be so cool. Just go outside. But what if you had to wear green to do it and you ain't had no green in your closet that day? You're just going to be hungry all day. Okay, never mind. Move on. All right. Lysosomes.
Oh, there would be a bunch of clutter and stuff, and nothing
Bunch of waste.
disposed of.
Right, because the lysosome is job. So if a mitochondria stops working, you don't need that non-functioning mitochondria in the way. So a lysosome's job would be to break that down, right? So you'll have a bunch of non-working things inside the cell. Also, you have a reduction in immunity because another way a lysosome functions, so if like a bacteria or virus gets into a cell, lysosomes are the organelles that fuse with phagocytes to actually break those things down. So you have a reduction in immunity as well as just a bunch of clutter in the cell. You know, kind of like when y'all don't clean y'all rooms up and start pushing stuff under the bed until it start falling from under the bed. Was my oldest son the only one that did that? Okay.
No, I'm doing.
You doing that right now? Centrioles. Right. No cell division or loss of cell division. All right. Centrosomes. A loss of cell division, right? But that wasn't yours, I don't think. I'm pretty sure that was not yours, but okay. Cilia and flagella. All right, there's a loss of movement. All right, so cells that depend on that solely for movement would not be able to move. And the plasmid. Bacteria would not be able to survive those harsh conditions. They would die at a faster rate. Cool beans, applesauce, and all that other stuff that we like to eat. All right, I still got 19 minutes and I'm about to use it. So have you already done page 103? I want to be sure.
No.
We did page 103, and I know we did page 104.
Oh, we didn't do 106 because you don't know what happened.
We didn't do 104?
No, we didn't.
All right, cool beans. I actually like 104. All right, let's do 104. This is the way this goes. You need your, no you don't. We use the boards on the last one. So here's the thing. I'm going to say a number and a function. We're not using the boards yet, but we will. I'm going to say a letter and a function, and then you are going to match that letter to the organelle. The top cell is a prokaryotic cell, and we know that because we can see something lying in the other stuff freely. I'm trying not to say the names of the organelles. And then the bottom cell is a eukaryotic cell that is an animal cell specifically, and we know because we see that there's some things that are not present. There's some things that are missing. Now, for the bacteria cell at the top, I want you to go ahead and put Juliet by number 1, and I want you to put Igloo by number 10. Those are two organelles that we don't necessarily discuss in here, and that would be your inclusion of storage granule and a capsule, okay? A capsule provides extra support, and an inclusion of storage granule is actually doing just that, is storing things. So 1 would be Juliet, and 10 would be India. So go ahead and write those down. So that means we won't be doing I and J because you already have I and J. But we will start with A. Now, I'm going to say the function, and you're going to tell me the organelle. All right, please put the boards down until we're ready to use them, Eli. Thank you. So, this organelle functions in movement or locomotion. It is a hair-like projection that likes to move in a windshield wiper-like fashion. Which number goes with alpha? 6. That's the pili or cilia. So beside number 6, you would write alpha, the letter A.
Oh.
All right. You see how we doing this?
A little bit.
I am going to say the letter. I am going to give you the function, and you in the word bank, you are going to find the organelle, and then you will write that letter beside the organelle in the word bank.
Gotcha.
All right. Moving on to B. This organelle also functions for movement or locomotion. It moves in a snake-like fashion. Which one is it? 9. It is the flagella. We trying to flag Ella down. Yeah, never mind. All right. See, I'm glad y'all didn't see that. This organelle holds the genetic instructions for the cell. It stores and transmits genetic information. What is C? Well, it's not an organelle, but it's, yeah. What is C?
2.
2. It is DNA. Please make sure you notice that that DNA is lying in something, and you can see it in that cell. That DNA is also circular chromosome. It's in a circular chromosome, which is different from how the DNA is in eukaryotes. They're linear. So in prokaryotes, you have a single circular chromosome. In eukaryotes, you have multiple linear chromosomes. All right, moving on to D. This organelle helps bacteria survive adverse conditions. It is a smaller packet of DNA. It is a smaller packet of DNA. What is delta? 7. It is a plasmid. That is a plasmid. Moving on to E. I don't know if you can tell, but it's pointing at those things that look like little dots. And their function, I'm going to go over all of them. If you miss one, I'll go back at the end. You got a question? It's fine. It's probably either dead or.
It's on 20. Can please
Wait, it looks
go.
like it's still.
It's still recording. It's just saying put it on low power mode. All right. Yeah. This one is pointing at the little dot inside of the cell, and its job is not really a dot. It's made of subunits, but its job is to make proteins. Its job is to make proteins. What is echo? 4. Echo is a ribosome. All right. Our next organelle, foxtrot, is the site of most metabolic activities. It's the site of most metabolic activities. What is foxtrot? 5. The cytoplasm. All right. Up next we have golf. Golf controls what enters and exits the cells by helping to maintain homeostasis. Golf controls what enters and exits the cell by helping to maintain homeostasis. What is golf? 3. The plasma membrane. And we already know that means that hotel must be the cell wall, which helps to provide extra structure and support for the cell. All right, now let's move on to the animal cell at the bottom. Notice there is a different word bank for the animal cell, and this time I'll be doing numbers and you guys will be matching the numbers to the letters. I am going to start with number 1. Number 1 is located inside of number 2, and its job is to make ribosomes. What is number 1? Hotel, that is the nucleolus. 1 is the nucleolus. Now, I said 1 is inside of 2, and 2's job is to control most of the cell's activities. What is number 2? That's alpha, the nucleus. 2 is alpha. All right, 3 is pointing to a thing that is located on 5, and the thing that is on 5, number 3's job is to make proteins. What is number 3? A ribosome, and that is delta. So 3 is delta. 4 is a sac, a membrane sac, a generic name for a membrane sac. And if I put different things in it, I would change its name. But right now, I'm just going to say it's a generic name for a membrane sac. What is 4? It's what? India, vesicle. Remember, I have to put something in it before I can call it a lysosome. That's just a generic name. That's a vesicle. All right, that's India, vesicle. 5 has those ribosomes on it, and its job is to transport the proteins that the ribosome makes. What is 5? It's echo, the rough ER. All right, 6 is going to sort, modify, package, and ship any proteins or lipids that enter into it. What is 6? Kilo is the Golgi bodies. If you don't know the letter, you can definitely say the name of the organelle. All right, 7, you have to look down towards the bottom. 7, it looks like a little filament or tube. 7's role is to help give that animal cell structure and support. Its role is to help give that animal cell structure and support. What is 7? What is it? Mike, cytoskeleton. Remember, animal cells do not have cell walls, so instead they use a cytoskeleton. Cyto means cell, so it literally means the cell's skeleton. And what do skeletons do for us? Gives us structure and support. All right, moving on to number 8. Number 8 is an extension of number 5 without the ribosomes, and its job is to synthesize lipids as well as detoxify chemicals. What is number 8? Juliet, the smooth ER. Moving on to number 9. Number 9 is an energy conversion organelle that converts the stored energy in food to the energy of ATP. So it makes ATP. What is number 9? It is golf, the mitochondria. Number 10 is one of those vesicle things that we talked about earlier, but it actually has digestive enzymes on the inside of it so that it can help clean the cell. What is number 10? Foxtrot, that is the lysosome. Number 11, I don't know if you can see it, but you see where you have those two number 9s and the number 4. Number 11 is written right there, and it's talking about all that stuff that fills the space between the nucleus and the plasma membrane, helps hold those organelles in place, and is the site of most metabolic activities. What is number 11? What is number 11? Charlie. Now, make sure you take note of the fact that cytosol is just the liquid itself. And when we say cytoplasm, we're referring to the liquid and the organelles. So those terms are used interchangeably, okay? So the liquid itself, we just call it cytosol. But if I'm talking about the liquid and the organelles that's in it, we call it cytoplasm. You'll find those words used interchangeably. So when you see cytosol, think cytoplasm. When you see cytoplasm, think cytosol. All right, number 12 is another one of those vesicles we talked about, and it is used to store water, food, waste, salts, and things like that. What is number 12? Lima. It is a vacuole. 13 is used to aid in cell division, found only in animal cells. What is 13? November. Centrio. It is November, which is next month. Coincidentally, my birthday is in November. All right, November 8th, I'll be 40 fun. All right, so moving on. That's 41 for those of you that don't know. I'm already 40. All right, 14 controls what enters and exits the cell and helps maintain the